dear students, how are you all? I hope you all are fine. In the today's video, we are going to discuss about class 10 topic, and we are going to talk about cell division. As you know that the body of all kind of individuals that present in our planet, any kind of living individual, the body is only made up of cells. Some have prokaryotic cells and some have eukaryotic cells. So in today's lecture, we are going to discuss about how a cell get divided into two cells. So basically, we are talking about cell division. Generally, as you know that the body of individual is made up of cells. So cell size is generally small when a cell is going to be born. So new cell is always look smaller. And after eating some kind of food and after doing some anabolic activities, after getting some food from surrounding, the smaller cell size growth will be happening. The first phase of the cell division or the cell cycle is going to be called by the growth. Because of the growth, the size of the cell is going to be increased. And here the formation of cytoplasm will be happening along with the formation of organelles, and here mitochondria is also present over there along with the nucleus. So every kind of things that present in the cell organelle, cytoplasm, everything is going to be increased by the process of growth. And when a cell so growth, it gets converted into mature cell. So after maturation, mature cell is just going for the division. Mature cell always for division. And because by the division, the single cell is get converted into sometime into two daughter cells and sometime into four daughter cells. So because of the division, the smaller cell is get converted into daughter cells over there. So what will be happening? Here two kind of things generally happen with the new cells always. The first thing is a growth, and the next thing is division. Growth and division is collectively called by a cell cycle. Cell cycle means every cells that present in our planet first show birth, and after that smaller cell get converted into larger cell with the help of growth. So growth will be happening and later division will be happening. Because of the division, cell number is going to be increased here. So we are going to discuss about in which manner the growth in the cell will be happening, and after that in which manner the cell division is happening over there. So first we have required to discuss about the cell division. But it might get converted into larger cell, and after that what will be happening when a cell is starting their division phase? The first thing will be happening is called by a name cell cycle, and cell cycle is just made up of two kind of phases. First phase is called by a name interphase, and after the interphase generally tell about the rest phase of cycle, rest phase. And in the rest phase, always growth, only and only growth will be happening inside the cell. So here a cell is growing, size increment will be happening, protoplasm increment will be happening, organelle growth will be happening inside the cell, and because of that, cell cycle is going to be increased. So first phase is interphase, and after that, the next phase is going to be happening is called by a name M phase. M phase is also known by a name actual division phase. Division phase. So in that phase, the larger cell, mature cell that have nucleus and that have mitochondria is going to be divided, and after that the formation of two cells will be happening. So cell cycle, the life cycle of any kind of cell is just pass through two phases. One is interphase and the next one is M phase. Interphase is generally divided into three phases. Growth is generally considered in the three forms: Z1 phase, S phase, and Z2 phase. And after that, what will be happening? The actual M phase will be coming over there. So generally, any cells want to divide, so that must have required to pass through Z1 phase, and after that S phase, after that Z2 phase. I just want to draw these four phases on over the cyclic representation. So I am showing the cyclic representation or cell cycle over there. I just want to tell कि some phases, as you know that for cell division preparation is required. So for preparatory phase, here a preparatory phase is over there that is called by a name interphase. This whole zone is to just tell about the interphase. Interphase always can belong inside that the complete cell growth will be happening, organelle growth will be happening. The time taken by the interphase is 95%. Along with that, the actual division phase that is represented by M phase is only take 5% time. So 5% time is just for the M phase and 95% time is gone for the interphase. And interphase here is divided into three phases. One is called by a name Zebul phase. Zebul phase is also called by a name growth phase or gap phase. Gap 1 are also called by a name growth 1. In G1 phase, the cell always take food from outside, and because of the food, the cell size is going to be increased here. Cell size increment will be happening. And for increment of cell size, we have required to form protoplasm. So first here the formation of protoplasm will be happening. Along with that, organelles also show growth. So organellar growth will be happening in this case. Organellar growth, protoplasm increment. Along with that, here the formation of ribosomes take place. Along with that, here RNA formation take place. But in this phase, G1 phase, never DNA is going to be formed in this phase. So DNA synthesis is never happen in this phase. Here protein synthesis also take place. And all kind of component is going to be formed that is required for a growth of a cell. But in G1 phase, our DNA synthesis will be not happening. So after that, the next phase is fall into existence, and that is called by name S phase. S phase, the full form of S phase is synthesis phase. Here, the synthesis of some cellular material is going to be happening, and this cellular material is called by name DNA. So in this phase, only DNA synthesis will be happening, that is called by name DNA replication. Means the amount of DNA, if a cell, a smaller cell is present here, so that's so growth, and after that the cell size will increase over there. And inside the nucleus, you know that DNA is present over there. So in this case, if the amount of DNA is 1 kg or 1 picogram, then after this phase, the DNA amount is going to be increased, and that will become 2 pg, 2 picogram. The amount of DNA is going to be duplicate here, so that replicate here, so that it is called by DNA duplication or DNA replication. It also called by duplication. Duplication of DNA, the whole amount of DNA is going to be double here. Along with that, inside the cytoplasm, a special organelle is present in animal cell that is called by a centriole. Centriole also show division in case of the S phase. So here centriolar division is also happening, centriolar duplication. And here formation of only a special protein is happening, that is called by the name histone protein. Histone protein also formed in this phase. So this is the second phase of the interphase, and inside that only DNA replication will be happening, or only centriolar replication will be happening. Later another phase is also present over there, that is called by a G2 phase. The full form of the G2 phase is gap 2, or it also called by a name growth 2. Second time again the cell start growing. Here a cell is present over there, and later it also show growth, and because of that this whole cell is just converted into mature cell. Along with that have double amount of DNA. So inside that protein synthesis will be happening. Here RNA formation will be happening, but DNA formation never take place in G2 phase also. And here the cell that present over there, in this case when we started the cycle, this is new cell, but the cell that obtained after the interphase is just known by a name mature cell. So here mature cell is present over there, and mature cell is just fall for the M phase, that is called by a name actual division phase. Inside that this cell, this mature cell pass through two kind of stages, that is called by a name, first one is called by a name karyokinesis, and the later one is just called by a name cytokinesis. Inside the karyokinesis, nuclear division will be happening. In case of cytokinesis, here cytoplasm will be divided in this case. So let's discuss about M phase. I am telling that M phase is the actual division phase that is divided into two phases. One is the nuclear division. Mature cell first show nuclear division, and nuclear division is called by karyokinesis. So first karyokinesis will be happening, and later in the second phase of the M phase, here the cytoplasm division will be happening. Cytoplasmic division. And because of the cytoplasmic division, the mature cell is get converted into two cells. So here cytoplasmic division will be happening, that is called by name cytokinesis. Cytokinesis will be happening. Nuclear division is a complex process. It is not a simple process. It is a complex process, and it always falls through four phases. So nuclear division and karyokinesis have four phases. Inside that, the preparation of this cell is going to be happening, and inside that, many changes will be happening inside the nucleus and chromosome, and because of that, cell is prepared for a division. So first phase of the nuclear division is just called by the name prophase. Second phase of the karyokinesis phase, M phase, is called by the name metaphase. Third phase is called by the name anaphase. And the last phase of the karyokinesis of M phase is just called by the name telophase. Prophase, metaphase, anaphase, telophase. Four phases will be there for the form here. In short, you can just recall like that PMAD. PMAD is the sequence of the event that is going to be happening in karyokinesis. So we will discuss about PMAD in the next slide. Here we are discussing about M phase. And M phase is generally happen in three ways. I told about that. And here we are discussing about mitosis cell division. Mitosis cell division. And inside that we are talking about karyokinesis. If you talk about the meiosis, the cell number, here the single parental cell is going to be converted into two cells, and never the chromosome number is going to be changed here. If a cell that present over there, that is diploid cell, means the chromosome of this cell is just present in pair. The daughter cell also have the chromosome that present in pair. Diploid cell, तो क्या करेगा ये diploid cells में बात करेगा. Or never the chromosome is not chromosome number is not going to be changed in this process. The chromosome number is always remain stable. Along with that, here what will be happening? Mature cell is going to be converted into two cells. And for that, we are talking about karyokinesis. The first phase of the karyokinesis, I said that is prophase. And after that, we talk about metaphase. Metaphase is the second phase of the karyokinesis of the mitosis. Later, the next phase is just called by the name anaphase. So we will talk about anaphase. And the last phase, that is the fourth phase of this karyokinesis, is just called by the name telophase. So we will talk about sequential phases. So first one is just called by the name prophase. So inside that, what will be happening? This is the cell membrane of the cell side. This is cell membrane. I am drawing the cell over here, and along with that, inside that, nucleus is present over there. So this is the nucleus, and inside that nucleus, you know that nucleotic material is present over there. That is called by the name nucleic acid or chromosomes. So this is chromosomes, and this membrane is just called by the name double membranous structure. Is the nuclear membrane. Nuclear membrane. Along with that, any cells that fall into prophase have always cellular organelles are always present inside that, and cells so very small type of all biological processes. So here what will be happening when a cell fall in prophase? First here the breaking of the nuclear membrane will be happening. Breaking of nuclear membrane. Nuclear membrane is going to be break here. Because of the breaking of both nuclear membranes, the whole chromatic material is just mixed with the cytoplasm. This is cytoplasm. So materials that present inside the nucleus is mixed with cytoplasm. Along with that, the chromatin thread that present in this zone is also show condensation. So chromatin thread that is later called by name chromosome also show condensation. The meaning of condensation is going to be dark. पहले क्या होता था that present only in form of thread, later it converted into rod cell structure that is called by the name chromosome. So here condensation in chromosome will be happening. Along with that, here nuclear membrane is going to be disappear, and along with that organelle also show disintegration in this case. But mitochondria and chloroplast never show disintegration because mitochondria is required because of the energy. if any cells want to division, then much energy is much required over there so that the breaking of mitochondria is never happen in this phase. So later, this is the prophase, and here what will be happening? Breaking of nuclear membrane, along with that condensation of chromosome happening. I told about that inside the cytoplasm, a special nuclear apparatus is present over there, and that is just called by a name centrosome. So centrosomes is present here. Centrosome first show division, and because of that, the number of two centrioles is present here. Later, it start moving toward opposite poles of the cells. This is the pole, and toward the pole, these both centrioles that present in the cell start moving away from each other to just reach at the north pole and south pole. And after that, here chromosomal condensation is going to be complete. Later, this whole chromatin thread is converted into chromosome. The next phase is just called by name metaphase. In the metaphase, all the chromosomes are chromatin material that present inside the cytoplasm of the cell that have integrated nuclear membrane and integrated organelles. It just called for the next phase, and in this phase, what will be happening? Chromosomes, the chromatin thread is just converted into chromatin thread is just converted into chromosome. Here formation, here I am drawing four chromosomes, and that have two chromatids. Here I am drawing a chromosome, and that have chromosome is just look like that, and you know that these are the thread of the chromosome, or arm of the chromosome is just called by a name chromatid arm. Chromatid arm, and the central portion is called by a name centromere. And here a disc-like apparatus is present over there that is called by a name kinetochore. So such thing is required to understand before the metaphase. And here what will be happening? Repulsion in the centrosomes will be happening, and because of the repulsion, these both centrosomes it just reach over the opposite poles of the cells. So here I am drawing the centrosome, and another centrosome is here. When a metaphase is just come into existence, such nuclear operators or cellular operators start forming such kind of special ester-like structure that is called by a name esters. It is just made up of microfibrils. It also called by a name spindle. This is centrosome. Two centrosomes is present in the cell when cell is in prophase, and along with that, centrosome also start formation of spindle. So here I am drawing spindles. Spindle just came out from centrosome and get attached with kinetochore of the chromosomes. Here the thread is coming out and get attached with the. Here what will be happening? The spindle fiber length increasing, and because of that, what will be happening? The chromosome get attached with this kinetochores. And along with that, some fiber are also present over there that move from one pole to another pole, and that is continuous fiber. Continuous fiber never attached with chromosomes. So what will be happening? All the kinetochore of the chromosome get attached with the spindle fiber that came from centrosomes. And here, this location is equator of the cell. Equator of cells, and this location is just called by a name. So toward the pole, centrosomes are present, and at the equator, chromosomes are present. This is the chromosome plate. Chromosome plate. In metaphase, a plate is forming over there, and that is called by a name metaphasic plate. The chromosomes are just arranged over metaphasic plate. So here, in the metaphase, the formation of metaphasic plate will be happening, and all the chromosomes that present in the cells it just get arranged at a single equator. It called by a name single metaphasic plate and single equator. All the chromosomes are get arranged over there, and this arrangement of the chromosome is just happening because of the spindle fiber that arises from the centrosomes. So here, arrangement of chromosomes on metaphasic plate is called by a name congregation. Congregation will be happening. After the congregation, in the next phase, the separation of this area will be going to be happen. This is just called by a name breaking of centromere will be happening, and because of the breaking of centromere, both the chromatids, here one chromatid, two chromatid, both are called by a name sister chromatid of a chromosome is going to be separate from each other. In the next phase will be happening. So I am drawing a diagram of the inner phase. And along with that, here cell elongation will also happening in this phase. So in the inner phase, the centrosomes are just present at the poles, and They are just breaking. Here what will be happening? This is the metaphase, and all over the metaphase, which thing is present over there? The chromosomes are present over there. And in case of metaphase, you know that spindle is just attached with the kinetochore of the chromosomes. So this is the diagram of the metaphase, and after that, here anaphase is just come into existence. And because of the anaphase, what will be happening? A pressure is just generated by these spindle fiber, and because of that, breaking of this zone will be happening. Here, breaking of centromeres will be happening. Centromeres. Because of the breaking of centromeres, both chromatid of the chromosome will just start moving away from each other toward the poles, toward opposite poles. So I am just drawing the next diagram, and inside that I just want to show that kinetochores, and here what will be happening after the breaking of the centromere, chromatid start moving toward opposite poles. So these are the chromatids, sister chromatids of a chromosome, both separated from each other and start moving toward opposite pole. In the next phase, the spindle length of the spindle again is going to be short, and because of that, the movement of the chromatid is just going upward. Here, the tail of both the chromatid in this phase always lie toward equator, and the center portion of the chromatid is always lie toward poles because of the sorting and because of the force generated in such a way. Along with that, this whole structure that I am drawing here is called by the name spindle fiber, and length of the spindle fiber is decreasing. Again, the chromosome or chromatid thread is just moving upward. So this is a dynamic process, and in this whole phase, the both chromatid of chromosome are just reached at the opposite pole of the chromosomes. So such kind of diagram will come over there. In the telophase condition, what will be happening? The dissolution of whole this spindle fiber will happen, and these whole structure that I draw over there is chromatid. That chromatid arms are just get gathered over the poles, opposite poles. So here I am drawing over there. Here the breaking centrosomes is present over there along with centrosome. Here chromosomes that have single chromatid, so four chromatid I am drawing here, and another four chromatid, sister chromatid is just present at the opposite pole. So such kind of diagram is observed in this case. Along with that, in this case, such kind of structure show decondensation, and because of the decondensation, they again converted into thread-like structure. So such kind of diagram is just observed over there. So this structure later so decondensation, and because of that, they just move here and there inside the cytoplasm. At the finishing at the end of the telophase, here the formation of nuclear membrane is also telophase. So two type of chromosomes are present over there. That chromosomes is just called by name daughter chromosomes. Daughter chromosomes. And the nucleus that generated over there is just called by name daughter nuclei. This is daughter nuclei. The product of the telophase is the formation of two daughter nuclei here, and along with that, here in this phase, cytokinesis also going to be happening in this manner. And here the formation, reappearance of nuclear membrane is happening. Reappearance of nuclear membrane. Along with that, inside that, what will be happening? The formation of new organelles also happening start. Along with that, here formation, first the formation of ER will be happening. Later, ER is converted into Golgi body. The formation of Golgi body, lysosomes will be happening. As you know that mitochondria is present over there. So inside the parent cell, a single mitochondria is present over there, and in case in the last mitochondria are also able to make copy by itself own is called by, and then here the two mitochondria is present over there. So this is the end result of the telophase. Everything that telophase is also called by the name reverse prophase. So all the things that happen in prophase, here the dissolution of all kind of cellular material will be happening, but in this case, what will be happening? Reappearance of all kind of cellular material will be happening. So this is the whole things that is related with the karyokinesis, mitosis. Later we are going to talk about the next phase after the karyokinesis is called by name cytokinesis. Cytokinesis is a process that happen that start from mid anaphase and completed after telophase. Cytokinesis never going to be happen just after telophase. It always starts from anaphase when elongation in the cell will be happening. So from this zone, the cellular division is also happening here. Because of that, the cell size cells is just looking this manner. So we are going to talk about cytokinesis. So here the next thing that happens in the cell division is cytokinesis. Cytokinesis is generally did by a cellular division. As you know that in case of telophase, the nuclear materials are chromatin thread are just arranged here, and here it is surrounded by double membrane nuclear wall. Here also the reappearance of double membranous nuclear wall will be happening, and along with that it surround here the chromatin materials. Here in the if you talk about cytokinesis or cellular division is start from anaphase. It always start from anaphase, mid anaphase. But the completion of cellular division is always happen after telophase. After telophase. So we are going to talk about that. So this is the cell that present in the telophase. And here the formation of boundary will be happening, and because of that the single cell is going to be converted into two cells. So in case of if we talk about cytokinesis, so cytokinesis in different individual is going to be happening in different way. So we are going to talk about two kind of cytokinesis. Two kind. The cytokinesis that happen in animal cell, and another cytokinesis that is going to be happening in plant cells. Animal cell cytokinesis and plant cell cytokinesis. In case of animal cell, as you know that the body of the animal cell is just surrounded by cell membrane only. So here what will be happening? The congregation of the membrane will be happening. The membrane that present over there, cellular membrane, so such kind of pitching. And because of that, the cell membrane is just moving inside, and later it just meet in such a way. So here what will be happening from outside, cell membrane start moving to outside to inside, and such kind of structure here rotation will be happening. जैसे कि हम बॉलून को लेकर के ऐसे रोटेट करते हैं, such kind of division will be happening, and because of that, the pinning of both the membrane will be happening, and so that cellular membrane is just moving inward, and such kind of division by that, the formation of two cells will be happening. It's just called by a cell formation. Cell formation. So in case of animal cell, the cell division or cytokinesis is followed by cell formation. The formation of cell follow is happening from outside to inside. So this kind of centrifugal, this is it's just happening like a centripetal manner. So cell formation always happening in centripetal manner. Next thing we are going to talk about plant cell. As you know that the plant cell is just surrounded by the hard covering. It's called by name cell wall. And after the cell wall, cell membrane is present here. So this is cell membrane. In case of plant cell, this boundary is hard. Cell wall is hard over there, and because of that, the pinching is not happen here. Cell formation never happen in this case. Here what will be happening? The two daughter nuclei is present inside the cell, and in between that, as you know that here endoplasmic reticulum, Golgi apparatus is present over there. Endoplasmic reticulum and Golgi apparatus mix with the cytoplasmic reticulum, and after that formation of a body will take place, and that will start from the center location. And this structure that start moving from center to outward is just called by name plasma. Later phagoplast is just combined with the material that present in the cytoplasm, and here the boundary formation will be happening. Here the generation of new cell wall will be happening. Phagoplast is just converted into new cell. So in case of plant cell, because of the presence of the cell wall, phagoplast is just going to be formed. A wall-like structure is just formed between two daughter cells, and later the cytokinesis of the plant cell will be converted here. So phagoplast always start forming from center to outside. This is called the centrifugal manner of cytokinesis. So here we talk about two cells. So in general, if you talk about mitosis, so from the mitosis we got a mature cell is just converted into two daughter cells. And nuclear number or chromosome number is never going to be changeable. If you talk about the human cell, the two in human cell have 46 chromosomes. The daughter cells that form by mitosis also have same number of chromosomes. Daughter also have same number of chromosomes. These both cells have 46 and 46 chromosomes. Generally, if you talk about the mitosis, the significance of mitosis. So mitosis helps in cell growth. Because of that, the cell number is going to be increased. So it is used for cell growth, cell number increment. It also helps in repairing. Repairing will be happening. Bound healing is also happened by this process, and such kind of thing will be happening. Regeneration of our skin is also a part of